Hello. Welcome to the next talk of the BioCore Winter Conference. This talk will be given by Clockwork Show. Yeah.
Just making sure that my audio is like good and you can hear me and all of that fun stuff.
Yeah, I can hear you.
Perfect. Am I loud? Am I quiet? Because I'm on this stupid board thing, so I have to like manually control my audio. Cool.
Audio is perfect.
Thanks, friend. All right. So that puts us right at the start. So I'm just going to jump right into it, right? Oh, great.
Go ahead.
Awesome. All right, so I want to ask everyone to forgive me up front because, as you can hear, my TA is here in the room with me, and that's just going to be something we have to deal with. So if you hear cat noises, I don't know what to say. We're out here in LA in the middle of lockdown, so this presentation is going to come to you from a very crammed space due to the fact that nobody in this state can step outside anymore. Moving on. So, after all of these really exciting and nutrient-dense presentations for the majority of the BioCore Winter Conference, finally the liberal arts majors are here. So, we got bold fonts, we got pretty colors, we got flashy animations, we got a 2.4 GPA. We got everything you need to take you through something that is not quite as objective as everything else you're going to see during this conference. So we're going to go through the fundamentals of life sciences communication. So definitely the stream's still running. Awesome. I'm doing this off of an animation rig. I'm just trusting the Discord gods to let this happen. If this falls apart, I mean, I'll be able to cut something together with the audio later on. So we're going to go through this as best as possible, but I'm really excited to take you through the fundamentals of life sciences communication. Main thing I would just like is if we can keep all of our questions typed in the lectures conferences room, just to keep it as focused as possible, because unfortunately when you're talking about communications, especially science communication, the answer to every single question is going to be, it depends. There's a lot of context here that we're going to have to just race through. So to get you through that, let me just take you through what we're going to be talking about today. So first of all, number one, why do we, mostly a group of undergrads and graduate students, why do we even care about science and science communication? Number two, how do we do that science communication? It all depends on context. How do you level up an audience through your context? How do you make sure that you keep the ideas and needs of your audience in mind? Part three, unfortunately, we're going to have to talk about branding because I'm sorry, that's just the economy we live in. Part four, hopefully we can take those previous two elements, put them all together to help you figure out the right message for the right person, for the right time, for any presentation, video, awkward dinner conversation that you, a life sciences professional, will eventually have to have. Part five, we'll kind of tie it all together. And then part six, hopefully we'll have time for questions. I do have a hard out at about 5 minutes before the hour. Just to make just because like we have a 3-month-old, so it's just one of those things where I have to go back to baby duty as quickly as possible. So forgive me for having to be quick here. So let's just jump right into it. Why science communication? Main thing, I just want to also say hi. My name's Peter. I am a science communications professional at this point. I probably should have introduced myself, but now that we've gotten like the bones of the talk out of the way, I have been everything from a science teacher in South Korea to a science presenter in Western Australia and everything in between. My career has spanned, checking my watch, 4 continents at this point. In addition to that, I've also been a marketing professional for the New York tech scene, as well as now a freelance biomedical animator out here in Los Angeles. So good morning from sunny California. Good morning from lockdown. So with that really kind of long career, I started off as a biology major at a liberal arts institution in Pennsylvania and ended with a writing degree, thinking that medical writing would be the ultimate way to sort of like kind of combine my goofy personality with my sort of like bad lab skills. That has kind of turned into what I'm working on now. I am aware of BioCore because I run the YouTube channel This Glorious Clockwork. And by the way, if you're here in this talk right now, you are legally obligated to subscribe to my YouTube channel. I'm sorry, I didn't make this law, and if you have a problem with it, contact your congressional representative, parliamentary person, whatevs. Really strongly encourage you to subscribe there. Mostly joking, but that's mainly what I'm working on right now, in addition to a lot of biomedical animation projects for other YouTubers, as well as just like lots of other small projects to sort of like, weather the storm of COVID. So my main goal as a science communicator is to get people interested in and excited about biochemistry, it's hard. It's a little difficult. You're taking one of the most advanced and most difficult to understand parts of life sciences and trying to bring it down to somebody who's just like hanging out on YouTube. It's a really awesome and terrible nut to crack, and I'm having a lot of fun doing it. And I'm going to kind of take you through everything I've learned in the past year of doing it, as well as, oh, yeah, did you know? Really? Anyway, I'm just reading the comment. I'm going to just move my screen's around a bit, so I'm not reading the comments as they come in. I don't know why I'm doing that. Never read the comments. Just focus on your talk. So I'm going to take you through a lot of very specific examples specific to biochemistry, but my goal with this conversation that I'm going to have with you today is trying to give you the fundamentals of life sciences communication so that you can apply them to telling people why it's interesting that you're surveying a particular species of deer in Western Pennsylvania, or whatever it is you're doing in your lab right now, or whatever it is you're trying to do as you're trying to teach a particular branch of life sciences. Science communication is more important than ever, especially in these times. I'm not going to go into that too terribly much, but why do you, odds are an undergrad or even a high school student, and if you're a high school student, how dare you? How dare you even be here? I feel so ancient. Why do you even care about science communication? Because like when we think about science communication, we think a lot about like Bill Nye or Hank Green or people who are like doing very popular things on YouTube and or TikTok and or anywhere else. But eventually, this is one thing that I disdained as a lab person. I just thought when I was a bio major, I'm sitting there in the lab, I'm a pipette jockey, I'm PCRing everything within an inch of my life. I thought, just do the lab work, that's what matters. But no matter how long you study, you eventually have to explain your findings to somebody who's not a scientist, and you have to eventually convince people that your research deserves funding. And so a lot of your life is going to come down to these fundamentals. A lot of like the angle of your academic career will come down to this. And if you find a way to tell your story as a life sciences professional better, not only are you going to, air quotes, advance your career or make a cool personal brand, but you're also going to better understand your own place in your own personal story as you develop as a life sciences professional. Because it is pretty amazing how much you learn as a biologist, like just going from zero in Bio 101 to all the way in your senior year when you're like defending a thesis or going into grad school. It's absolutely incredible the amount of information the biology process crams into your head. So it's amazing. If you can tell yourself that story, you can have a better understanding of everything else. So let's get into the only part of this talk that matters, and that is context is everything. I have been everything from a science teacher to a marketing professional, so I have had to talk to so many different kinds of audiences. And the issue when you are a life science professional is that there's a lot of context that has already been laid for you within the medical field. So whenever you talk to, if you're a biochemist and you're talking about an enzyme, somebody like, well, how does that enzyme give you cancer? If you Google anything about biology, Google will tell you have cancer. If you even just think about it, the back of your mind will say, it's cancer, you're going to die. So what you have to do as a life sciences professional is really essentialize everything you're doing to try to get away from the kind of medical existential aspect of it. This is one of the chief sort of efforts you have in one of the chief issues you're always going to have as a life sciences professional. So how do you like get people through context? The issue in life sciences, particularly, like in physical sciences, it's just like, yeah, I do a thing and hopefully it doesn't blow up. But for us, we only have really two options. You can either tell somebody something super accurate, like yes, when you take aspirin, serine 516 gets acetylized in the COX-2 enzyme, which is, what did I even just say? I had literally like memorized that in like third year bio, like to this very day. It's just stuck in my head. You can be accurate, but it's so dense and hard to understand, you may as well have just spoken non-human language. Or you could say something that feels clear, but ultimately is misleading. Like you can say aspirin breaks your enzymes, and that's why you don't feel pain anymore. And that's no good, because what you want to do is you want to bring people to your understanding. And so one of the most important principles in life sciences communication is that you have to do things incrementally. And so one thing I want to help you do and help you think about is how you can shave your ideas down to their most essential shareable details without getting into misinformation or without getting into like a weird meme space. Like when I was a little like lab jockey, when I was pipetting, my main job was using VEGF on Podocarina, which is a kind of little hydra-looking guy. We were trying to turn Podocarina into a model organism for studying VEGF, which is the vascular endocrine growth form factor. Don't tell my lab boss that I forgot the word factor in there. How to use these little hydra-looking guys as a model organism for figuring out tumor growth and how to suppress VEGF. Because if you suppress VEGF, maybe we can figure out a way to mitigate tumor growth. It's a whole thing. And I would, go around and I would say, yeah, I'm giving jellyfish cancer, because I was 19 and dumb. And that doesn't help anybody. But it's really exciting thinking about how we can model tumor growth within within a literal jellyfish that has very little like relation. Our evolutionary branch branched off billions of years ago, and we can still use biochemistry to figure out our own tumors with these guys. So the main tension is always going to be, how do you make your ideas and work sound interesting, but how do you do that in a way that you can get people up to an actual understanding? And so to do that, I don't want to be in slides forever. Let's go into the actual meat of this. What are we doing? I'm going to live in Pymol, because if you're a biochemist, this is your house. Or at least if you're like a biochemical academic. And so I just want to show you how to level up somebody through an understanding here. So this is a surface scan of a human enzyme. This is COX-2, cyclooxygenase 2, which makes prostaglandins, which is very important in the pathway to inflammation and feeling pain. This is the enzyme that gets inhibited by aspirin, ibuprofen, naproxen. This is what we kill for pain mitigation. But if I just have this here, I'm like, yeah, this is your, like, I can't just say this is your pain enzyme or whatever. I can't just say, hey, this is, because, or first of all, what is this thing? Like, this is a surface scan. Like, I'm looking at the surface of the protein here, but you don't see that you have two chains. You don't see anything. And so you have to do levels of abstraction. So level 1 in all of biochemistry, we're going to look at the, boom. Oh, sorry, my bad. We're going to go to scene 7. Boom. And so what we do is we take away the surface scan and we just show the secondary structures. This is primarily made up of alpha helices. We see that. But then, like, again, if I bring this to a lay person, I'm just looking at a big pile of spaghetti noodles. Like, literally, I did this once as a young person, and somebody was like, yo, it's flying spaghetti monster. And so that doesn't help anybody, because this isn't, like, how do I tell you, look at this thing and you'll know that this is how aspirin works. So we go a little bit deeper in. We go to the active site of the enzyme, and Pymol's really good at this. So we see the substrate here. I'm not going to try to say it because I forget how to pronounce arachidonic acid, aranciodo. No, I'm not doing it. You're not going to catch me out on this. But again, this is just a bunch of pretty colors. Like, what does this mean? Like, so within Pymol, you've got a lot of codes that can show you the actual active site of any particular enzyme. And so now we're starting to get down to the brass tacks here, because we're seeing hydrogen bonds. We see this tryptophan here. We see the substrate. We see this serine over yonder. We got a water molecule. Someone just posted the actual pronunciation. It's arachidonic acid. Correct. At least that was correct back in 2009 when I was learning how to do this. And so this is good, but and so what I did was, and this is how you do this in real life, you go from, this is what it actually looks like, to, this is a representation of what it can look like, down to the actual details you need. And you use the context from one scene to get them to the next scene. It's all about scraping away detail. You want to get down to the essential facts as possible. Why do people care about this, though? Like, it's cool that there are these hydrogen bonds, but why does this matter? It matters because we zoom in to this particular serine. This is serine 516. When aspirin comes in, it's going to acetylize this. It's going to turn this one oxygen, this one OH functional group into an acetyl group and make the entire enzyme completely worthless. And so you see how this particular serine is extremely important because yada yada yada. It has this particular reaction with with that substrate. That's a really fast example. This is literally me just glossing over 45 minutes of detail, and it's cool to do this in PyMol, but how can you do this in your real life? Like, I understand you might not live your life in biochem. You might not have these kinds of essentializing details. You might not have an awesome program like PyMol, which is free, ? It said I've had 25 days remaining for my entire life, so I don't know what's going on there, but I'm here for it. You might not have free software like this, or something as amazing and free as the Protein Data Bank. You might, have to figure out a different way. And so let me take you through how I took the exact same process here and essentialized it with something that I created. So we're going to go back into PyMol. I'm going to go action, orient, no. I'm going to show my surface. What are we looking at? Right? If you're looking carefully, you can kind of almost understand where I'm going with this. Boom. This right here is just a free model of, it's the first hemoglobin I found. This is recombinant human hemoglobin. This is what it actually looks like in real life. Again, this is the surface, so you're seeing some of the oxygens and nitrogens in these amino acids. Kind of hard to understand, extremely colorful. We're good with it. Let's hide the surface again and see the structure. Oh, I want to also show the cartoon here. PyMol's fiddly, y'all. I don't know what to tell you about that. All right, so this is an accurate representation of hemoglobin, which is the protein that carries oxygen in your blood, so on and so forth. If you use this, obviously it's cool. Again, the main issue with talking about proteins is it's always going to be the flying spaghetti monster. So how do I find something that's actually valuable for an audience here? So I'm going to go, with biochem, it's just playing around in PyMol until you find something that makes sense to you. And for me, it was going back to the surface scan, please. I'm going to show surface one more time. Show surface, thank you. And this view, all of a sudden I see that all of those spaghetti proteins, all of those alpha helices, make these happy little pockets for this guy, my good friend, the heme group. So you got your iron in there, you got your ball and stick model for the heme group, and all of a sudden you understand that hemoglobin is a 4 subunit protein that basically exists around these 4 heme pockets. 4? Yeah, it's 4. And so what you do there is you abstract it out. So what does hemoglobin do? In a certain percent of O2 and a certain pH and a bunch of other factors, it holds onto oxygen, lets it go when pH goes down, so on and so forth. But what you do is you take those helices and you make them into something very small. And so what I've done here is, instead of living my life in spaghetti land, I'm living my life in something that's a little bit easier to understand. Like, I am communicating viscerally. There are 4 subunits. There's 2 different kinds. You got your alpha globin and your beta globin. Now, alpha's red, beta's blue, because of acronyms. And so right here, bam, 9 seconds, we all get what's going on here in hemoglobin. We understand that in a high percent O2, hemoglobin is more likely to bind with O2 at its 4 heme groups. And then as pH goes down and O2 percentage goes down, it's going to be more likely to hemoglobin. Heme itself does not bond with carbon dioxide. I should be very clear about that. It just associates it with a little bit better. But this is how you can start really teaching biochemistry, how it's, instead of just this thing to memorize, it's a dynamic system. Hemoglobin, rather than just knowing that it has oxygen and has to drop it off, it will drop off oxygen where your pH is lower, i.e. where your muscles are doing the most work and there's a CO2 buildup, and that pH of that the pH of that blood is lowering to the point that hemoglobin pops off, and it becomes this beautiful system, not just this thing to memorize. And this is how I viscerally began teaching biochemistry on the YouTube level. This is still a little bit too abstract, though, and so you have to have an intermediate step. So what I did was, instead of just having this abstraction, I need to explain it. I have to get from pretty colors, wait for it, gotta go back to this guy. So how do I go from here? Where'd it go? Here to here. It was right next to it. I went too far. So that's the main question. So what you do is you have to be able to talk about very quickly the actual accuracy in what you're trying to say. And so you go from talking about the 4 subunits, yada, 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 to, oh yeah, I did do it right. Awesome. I can't believe I remember this. To zooming in on a single alpha helix to make sure people know what an alpha helix looks like. So we have the abstraction here. And then we zoom in and we get the actual structure. And so you see you have one backbone, one backbone, one backbone. We're finally getting to some R groups. I haven't done a very good job. This is aspartate, but I made oxygen white because I was stupid back 6 months ago when I started this project. So one thing, too, to always keep in mind is that pretty colors are cool, but maintaining scientific accuracy is cooler. So you see that what I did was I made hydrogen red and oxygen white when it should be reversed. And that's because I'm stupid. That's the main answer to any question you have here. It's because I'm not a clever person. So we go through, so again, and then you really try to cement the idea that proteins are a sequence by demonstrating, that's an alanine, leucine, so on and so forth. And then it cycles back through because loops are really good in social media these days, right? So I want to take you through that actual process. So how do you figure out exactly how does this count as an abstraction for an alpha helix? And it's mainly just using the math of chemistry itself. Anytime you're working with math is also something you can use to make art. So we have a side view of an alpha helix here. The angle here is very precise, and so we know if we look at a ball and stick model of an alpha helix, we see that we have a backbone that goes down back, and then down this way in the front, down, and so on and so forth. We want to get to the point where we can demonstrate how these hydrogen bonds are forming to form this alpha helix structure, right? So what you do is you follow that exact same angle, and so you tell somebody viscerally that an alpha helix is a light thing in front, a dark thing in back, and this is a principle in art. Keep the lighter stuff in the foreground, the darker stuff in the background, and you get a better idea of what an actual alpha helix is. Now, I ran into problems here, though, because trying to explain biochem in 2D is bad. Don't do it. I don't know why I'm doing it, but it's the only way I can really bring biochem to the masses. And so this became a huge issue with my latest video, which is on ATP synthase, because if you look at ATP synthase, this might break everything. I don't know. If you can see this, it's a miracle. You have a very three-dimensional structure. You have the F0 complex of ATP synthase is literally a wheel. And so how do you demonstrate that? So what you have to do is you have to show it from lots of different angles. So that's a very chaotic animation that'll make a lot more sense when I've actually put it into YouTube as opposed to within After Effects itself. So what you do is you use two views. So if we look from where'd we go? There we go. We look at our F0 complex from the top, it becomes much easier to understand. It's literally a wheel, and it moves as protons come in. Actually, it moves because protons leaving causes a huge conformational chain in this particular, alpha helix. But the thing is, how do I accurately represent a helix from the top? Because if you don't respect the science, you can just make a circle and be like, I promise, it's what a helix is. So you go to the science and you try to find anything mathematical that you can use. And so if you go to an actual ball and stick model of an alpha helix, what you discover is, and what you learn, hopefully, or at least what you remember from biochem. I know most of us, when we go through biochem, we learn it and then immediately forget it because it's just like the most rote memorization part of biology. If we look here, we know that the angle from the top of an alpha helix is 60 degrees. We know that it adds up to, what is that? I forget the math here, but I know the angle from here to here is 60 degrees, which is awesome. Which is why having something like Adobe's Illustrator suite is awesome, because all Illustrator is vector art, and vector is just math. It's all math. So we go in here, I'm just going to build the top-down view of a helix in real time. That's what it's going to be eventually, but let's make it, honest to God, a real helix. So I have this, it's the exact right angle. I'm going to go to Object, I'm going to go to Transform, I'm going to go to Rotate, I'm going to go to 60 degrees, because I know that's the bonding angle. And then I'm going to hit Copy. Boom! I did it! And so then what I do is, you find the endpoints of each, and you stick it. Boop! No, don't do that. Should not have done this live. I should have just recorded this. What you do is you bond them up, and that is it. That's the bonding angle we need to play around with. This doesn't look right. This doesn't look like 60 degrees. I feel like it's 120, isn't it? I did my own math wrong, didn't I? I'm going straight to Wikipedia. Y'all can't stop me. Let's see. I know I have this right. What is it? Oh, it's a 100-degree turn. Right. Okay. no one make fun of me. No, this never happened. Delete this from the internet. So we go back here. It's 100 degrees. So let's delete that. Try again. So same idea. And like, maybe I don't need to do this fully, but you kind of get the idea. I'm going to, rotate. I'm going to do it 100. I'm going to hit, boom, copy. That looks a little bit more accurate. Join them up, and then rinse and repeat until you get something like this. And you always differentiate your subunits with a lighter and darker color. So what's my hex code there? Was that FEFTA8? Yeah, FEFTA8. And so I know what you're thinking. You're thinking, but Clockwork, how do you find the pretty colors that you use? You must read so much about color theory. I don't know anything about color theory. Once again, this is all down to just the mathematical relationships between colors, as told to me by the Adobe color themes. If you are an undergrad student right now, odds are Adobe is going to give you their software for free. They're like any good drug dealer. The first use is free. And everything afterwards is, eventually you will have to pay for it. But while you're an undergrad, odds are you can get this stuff for free. Get that Adobe Creative Suite. Get into Illustrator. Use math to make your biology art. It's actually very easy. As long as you understand, you're just doing math here, you're going to be just fine. So what I do is I just pick a base color. That one's nice. Let's go into our Adobe color themes. I am going to, and then all of a sudden I have all of the mathematical relationships for all the colors ever. And so the entire Clockwork color palette comes from picking a blue, literally just building the hemoglobin model at random, picking a color, rolling from there. And that's essentially it. And so this is how I did it for biochem. And the amazing thing about biochemistry is that when you're talking about proteins, you're either talking about alpha helices like this or beta pleated sheets. And then you just kind of put those all together to make a tertiary/quaternary structure. Quaternary. Anyway, nobody needs to know how to pronounce quaternary. That's not my job here. My job is to take you through sort of these kind of SciComm fundamentals, right? And everything else is just these three relationships. The 100 degrees here, this is 60 degrees, right? And everything else comes down to all of that. And again, one thing I always want you to do too when you're breaking down essential details, this is me super zooming in to the F0 complex and that ATP synthase. That is 12 alpha helices arranged in a circle. We've got 6 of them right here because we only can show things hemispherically. Quaternary, right. Ooh. For a guy who talks for a living, I just can't pronounce anything. It's kind of bonkers, kind of funny. So when you're also trying to find ways to explain something to folks, you can play around with scale too. The main idea here is that you don't have to make something completely accurate. What I've done here is that this is the functional group in the F0 complex that a proton binds to when you're spinning around your ATP synthase and generating ATP. This is aspartate or aspartic acid, depending on your region or whatever. The main business end of aspartic acid is 2 oxygens, and like the negative charge is kind of hiding in here somewhere. Obviously, these 2 oxygen atoms are not this big. I mean, you've got a nitrogen, a carbon, a carbon, a carbon, a carbonyl, the whole backbone of another alpha helix here, and then these 2 giant oxygens. That's not accurate, but it gets people down to the essentials so that they can understand that, it's cool, there's this whole helix here, but the business end is the negative charge in aspartate. And that's what that's what you're always trying to do. Take people through the context so that they can understand the essentials. And so the scripts I write are extremely long. If we go to, I'm not going to go there right now, my ATP synthase video talks about ATP synthase for exactly 5 minutes of a 20 minute video. Like, it talks about the action of ATP synthase, because the most important thing is the context itself. It's not about talking your way through what are we doing? Where's my water? Oh, I didn't have it up, but don't worry about it. It's not just talking about like the rote memorization. It's not just talking about ATP synthase synthesizes ATP. It's talking about the conformational changes that power that. So this is still a little bit, what is it called? Inaccurate. But if you made this an actual wheel, you can see kind of the half channels where protons come in. When a proton pops off, it causes a conformational change, which pushes the whole wheel forward. And so that's why I made it this way. And this is some fancy masking. This isn't super dumb, but you'll see it in a video on Tuesday, hopefully. And so when I say level up somebody through that context, that's the main thing I want you to do. Find a way to, whatever you're doing, break it down to its most essential details, and then level people up through that understanding. Like half of this ATP synthase script is about water's ability to auto-ionize itself, and why that makes protons in your water, and why that causes a bunch of causes the whole context in which life on Earth is possible, because we do not exist without water's ability to auto-ionize. We don't exist without pH differentials, and all energy on Earth is just because some random chloroplast and or mitochondria is shoving protons into itself. That's it. It all comes down to that. You're solar powered, but your solar power is determined on life's ability to organize that solar power. Like, we're literally just giving a big ol' F you to thermodynamics by existing, and that's amazing. And that gets to a next point. Once you have your essential detail, try to connect it to something bigger. My most popular video so far is about Photosystem 2, and so the main issue when you're talking about Photosystem 2, it's not cool, do not do volume. I command you to be muted. Thank you. I don't know if this is going to show up. Let me know, blah, 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 blah. I mean, Photosystem 2 and the P680 complex in there, that's cool, but what's cooler and what really helps this stick in somebody's head is the fact that all atmospheric oxygen on Earth exists because of cyanobacteria. Like, billions of years ago, cyanobacteria figured out eating sulfur was, and I would like to point out to folks that, cyanobacteria were just like, hey, I don't really want to suck down sulfur anymore. I'd like to try something else. So they literally stole sunlight, turned that into oxygen essentially, created the ozone layer, which allowed complex life on the surface of Earth to actually exist. And that is, we're about to fast forward through about 10 billion years. Not 10 billion, 10 million, where is it? Yeah. What are we doing? Come on. You're killing me here. You're killing me here, bud. Anyway, so there we go. Like, the actual creation of the ozone layer itself. This was a terrible idea, just showing straight up YouTube. So it lets you think about an actual fundamental question. Did the Earth make life, or did life make the Earth? And it's kind of both when you think about it, because life on Earth's surface cannot exist without the ozone layer, and the ozone layer only exists because of the original cellular life that existed on Earth. So that way, it really sticks in somebody's mind. That video is really popular because I tied it back to space stuff. And so that's what you can always try to do. Go down to the essential details, get really drilled into them, and then connect it to a bigger picture so it sticks in folks' minds. Because this is the most important principle in all of science communication. The thing you need to understand is that you do not exist. Like, the incredible complexity inside your own mind, the story you tell yourself, all of that does not exist. Like, it is impossible for you to break the barrier that is your skull and bring it into the actual world. You have to communicate it. The only thing that exists is what you share with other people. And so when we think about human history, when we think about our own lives, what happens is that you only exist as a character in the mind of your audience. Like, I feel real right now. Like, I'm feeling the actual mechanics of talking to you right now, but my actual existence is in your head. Not to get too meta here, but that's the main game when we're thinking about communication. Nobody exists except in the mind of other people. And so when you're thinking about all of the research you've done, when you're thinking about the hours and hours and hours and hours you've spent in the lab, that's cool, but all that is memories for you. So how do you take those memories? How do you take all of this information, all of this work you have done, and essentialize it so it sticks in the mind in as many people as possible? And we do this with the most obnoxious exercise on planet Earth. We gotta talk about branding. I'm really sorry. I wish I wasn't this guy. I hate being this guy, but we have to be this guy. We have to talk about you and your personal brand as a science communicator. And so, very quick Venn diagram. Do you actually need a brand? Yes. Also, there is no escape. You cannot escape the economy that we are stuck in. No matter what you do, everything will kind of view it as a character, and that character is your brand. And so the main thing you need to be thinking about as a nascent science communicator, as a researcher, as somebody who's even just thinking about science at all, is thinking about the character that is telling people this story. Remember, when you are in any communication situation, there's three aspects to it. There's pathos, logos, and ethos. There is the actual ideas, that's the pathos. There is the logos, that's logic. The pathos is the whatever. I did not need to go into Socrates for this nonsense. There's the person telling the story, and then there's the story itself. And I'm loving the comments in lecture right now, because we're already getting into people making fun of this idea. Meme yourself into being a SciComm professional. I agree, but your personal brand doesn't have to be you just jumping on trends on Twitter. It doesn't have to be you just like being like, don't eat grass on TikTok or whatever. Your personal brand is just a character through which your ideas are viewed. And the cool thing about branding is that it's something that exists over time. So what you're doing... Yeah, exactly. And that's what it comes down to, is that you have to like actually build. So when we're thinking about your personal brand, it is a character through which your ideas are viewed. So that's the major. When you're thinking about this, you need to find a way to essentialize yourself. I love this. I'm going to actually jump into the questions. So a lot of the questions are, can someone explain what is happening here? We're talking about science communication. Why can't we be a nameless label on a lapel? Cue what mic you have. I don't really have a microphone right now. That's the problem. I'm talking into some kind of goofy lav microphone. But I understand this is where the conversation is going to break down, because it's the part of the conversation I hate the most. But it's one of those things where you need to take these kinds of principles into your own mind so that you can think about them over time. I'm not saying you need to be like flashy or an Instagram influencer or any of that nonsense. You need to be mindful of the character that people see when they are viewing your ideas. So let me take you through my personal brand. If you look at Clockwork, where we go? I always like cycle through the entire thing. If we go back to my channel, nice. Like, my personal brand is that I'm extremely Catholic. Just like the most Catholic person on planet Earth. Like, I really just kind of digested like Catholic teachings, and just think about the titanic forces that I am caught between good and evil. My whole deal is that I think I've done so many bad things, and like I'm just using biology as a means by which to say that I'm actually a good person. I swear, I promise I'm a good person. Just tell me I'm a good person. Anyway, and so if you watch any of my videos, you'll go through like some science, and then like some big like soul-searching-y nonsense where it's just like, you are a beautiful being made of light because you're made out of proteins. And that kind of works for some reason. And so when you think about your personal brand, you have to think about sort of the kind of forces that you have been, caught between in your own personal life and in your own being. You'd be surprised how you can essentialize your own character, and that comes from self-reflection. But a personal brand can just be that for whatever reason, you hate the amino acid serine. You just hate it. Like, it's something that's very like character driven. Like, if you just on Twitter are just like, God, I hate serine so much. Or if like you're talking about any particular meme and you bring it back to serine, you're just the guy who hates serine. And that seems very silly, but it's a little character quirk that helps you characterize yourself and stick in the minds of others. And so I stick to people, or some people, 1,000 people, because I just make biology seem like it's this like religious experience. Like, I literally called this channel the most self-righteous, obnoxious name, This Glorious Clockwork. Like, you are, ultimately made out of protein, so you're made out of clockwork, but that clockwork itself is glorious, yada, yada, yada. I'm thinking of just renaming my channel Life Just Works, by the way, so it's less obnoxious. Try to figure out any kind. And this is one of those things that you experiment with. That's one cool thing about social media that nobody ever tells you. Like, if you're just watching on social media, it feels like this like scary nightmare abyss that stares back into you. But for the most part, if you're just posting on social media, everyone's ignoring you. Like, if anything of the past 4 years has told us anything, it's that you can say anything and people are going to forget about it an hour later, which can be sad, but can also be very liberating because it allows you to do experiments. It allows you to, be very scientific in the establishment of your personal brand. It allows you to like test out meme formats that work or that don't. It allows you to figure out this communication strategy, because odds are I don't remember the tweets I sent yesterday. I don't remember anything I've done in the past week. That's mostly because I'm a new father with a 3-month-old child, and like my entire ability to process memory has been destroyed by sleep deprivation. But that's kind of how we are working as a collective species now, too. And so that's a great way for you to start thinking about the essentialized character of who you are. Try to find those little granules that aren't who you are, but are like very shareable. Main thing for me, I've taken you through mine. That's kind of the review. I'm good with that for now. And that's one of those things where when you're thinking about developing your own air quotes personal brand, it's one of those things that you're not trying to do right now. It's one of those things that you're trying to do over time. The ultimate principle in this new social media universe we live in, the ultimate thing that actually matters is the principle of incrementality. You're making one post today, you're going to make one post tomorrow. A year from now, you'll have 365 posts. What I'm saying is that gradually you will tell a story, and if you do it effectively, people will go back through it and use that story as a lens through which to see the world. Like, I am using my personal brand, Proteins Are My Religion, to make people care about biochem, which I think is something that's very important right now. I think it's very important to get people into the advanced life sciences because you're seeing a kind of STEM brain drain where a lot of like folks are being inspired by things like Elon Musk and SpaceX and like Tesla and whatever. People are going into more engineering and physical sciences when we're at a point of like systems biology, computational biology, bioinformatics, where like life sciences is about to become completely bonkers in the next 10 to 15 years, and we need as many life scientists as possible. So for me, my main thing is screw God, proteins are your new God, come be a biochemist, and then, build a system by which we can have personalized medicine and computational biology and AlphaFold and all that tomfoolery. Don't go work in Silicon Valley. Go become a bioinformatician, yada, yada, yada. Oh no, engineers in the chat. Well, I mean, biomedical engineers are awesome too. And if you're in more of the physical science, I mean, I get it. You're going to space. You're superior to me. It's all good. Don't hate. But that's the major thing when you're thinking about this, is what is that kind of overarching story you want to tell? And it sucks because you have to take that down to a single sentence and then, give it supporting details. And that is the ultimate principle. Once you put all these things together, you have to think about getting the right message to the right person at the right time. Thank God we're not talking about branding anymore. That section went way too long. And so this 4-step process is it. All you need is these 4 sentences and you're done forever. It's just that obviously this is a simple rules situation that has complex consequences. And so that's the main thing you have to always be thinking about, is that in life you're going to have simple rules that have complex consequences. And if you're thinking about the rules and thinking about the procedure and the process, you can build out so much from there. So, I mean, that's kind of like the whole principle behind math, right? Simple rules, complex consequences. Moving on, though. So, for anything that you as a life sciences professional need to do, you do it in this order. First of all, identify your target audience. It's cool that you're doing all this awesome research. It's amazing that you have all of these notes. It's great that you're about to... Oh, and by the way, this is for talking to a larger group of people, right? This is not for writing the actual paper of your research. This is not for writing an answer on a test. If you write like this in an academic setting, your peer reviewers will destroy you and you'll get a D in organic chemistry and then you will never get over it, ever. Anyway, that's not relevant to anything here, though. So when you are thinking about taking this away from an academic setting and bringing it to the rest of the world, first of all, identify your target audience and identify, very, very importantly, their attention span. Like, if you are doing something on YouTube, you have a lot more attention potential than, say, on Twitter. In Twitter, you have not just the 140 characters in which you are writing, you have the less than half a second somebody has to scroll through their feed and see what you're talking about. So maybe don't think about Twitter too much because that's very essentialized, unless you like really want to be like a big, like a super big deal or whatever. There's a way to work Twitter to your advantage, but you have to really like Twitter to do it. Twitter's not my game. I like to talk for long periods of time. I've actually been talking for 45 minutes. That's incredible. So when we are thinking about this, identify your target audience and then identify their attention span first. A great way to do this is just to sort of like think about the actual place in which this is going to live and think about all possible lifetimes of this particular piece of media, whether it is you're giving a talk at your school to a bunch of English majors for some godless reason, or you are, doing a poster presentation at sort of like a science fair scenario, or you're doing this on Zoom just to sort of like explain to your family what it is you do, locked up in a lab all day. So what do you guys know about carbon, so on and so forth? Identify your target audience and then determine to your best of your ability their level of understanding. And when you're in doubt, you're always going to play to the lowest common denominator. It's cool that there's a couple of PhDs in the audience right now, but I want to make sure that I am also speaking to people who are also potentially in high school and thinking about how to communicate themselves through this social media morass that we now live in. Because when I was in high school, this didn't even exist. There was no Facebook. Well, there was, but it was just for college students. Like I was in high school right on the edge of social media. Like back in the day, I was on MySpace. You kids don't even know. I'm such a grandpa. And it's a very good point in the actual chat. It's cool that you want to talk to the level of PhDs, but PhDs don't need your help to understand something. When you're sci-comming, you're trying to bring people from outside science into your scientific level of understanding. So always assume that people just. This is also a central tension in science communication because you want to assume your audience knows basically nothing, but you don't want to condescend to them. Like, so one thing I always do when I'm thinking about writing my scripts is assuming that everyone in the room is smarter than me about something that I'm not talking about. And that really works out. That helps you like write in a more empathetic way, because one thing that people kind of are used to from science communication is the sense of smugness. And when I say they're used to it, I'm not saying anybody in this room is smug or any particular science communicators in the world have been smug. It's just that's kind of how, unfortunately, despite science being a very, work pale existence when you're an academic, when you're doing research, you get kind of lumped in with like the air quotes elite of any particular social order. So people will assume, and so what'll happen is that people just kind of assume you're talking down to them. And so what I always do is just assume everyone's smarter than me. This can also be a problem. And here's the problem with talking about communications, because there is a but to every single sentence. There is do this, but, do this, but, do this, but, all the way down to infinity. While you're talking up to somebody, you don't want to come across as super unsure, too. You want to speak with conviction, because whatever it is you're doing, it's cool. The title of this talk is Everything You Do is Beautiful. I don't care how boring you might find your particular research path being. I don't care how mundane it is to you now that you've been in grad school for 5 years and you're just like living in your laboratory at this point. Whatever it is you're doing, a human being is doing it, and therefore it is gorgeous. And that is something that if you kind of bring that empathic if you're talking about that, if you're bringing that kind of empathetic perspective from your own writing, that is going to translate to your actual audience. So determine their level of understanding and talk up to them, if I could essentialize that as best as possible. And again, I really appreciate everyone's patience with how squishy this presentation is. Every other presentation during this conference has been, well, yeah, this is how the heart works, and we're going to go through every detail. For communication, it's, yeah, but maybe. Once you have determined their level of understanding and kind of figured out a way to talk up to them, get your context going and assemble an army of supporting details. And then the most important thing is repeat, repeat, repeat your main idea. Find 3 points throughout the actual thing. Oh, I was disconnected for a second, and now I'm back. We can hear me, right? We can hear me. We're hearing me now. I'm seeing a lot of typing. I'm feeling
You are
squishy.
audible.
Good. Hello, beautiful. Thank you. Did I actually? It doesn't matter. So, as I said, and I'm going to repeat it again because it's the most important thing. Repeat, repeat, repeat your main idea. Always find a moment in which you are really nailing home whatever it is you're talking about. In my videos, you'll find like 3 to 5 points where I say hemoglobin transports oxygen because of pH, or ATP synthase works because of different charges in water, yada yada yada. That's not what I actually say. I just kind of forgot what I was saying before. Always find a way to like nail down your main idea at the very beginning, kind of in the middle, and then at the very end of whatever piece of information you're giving. Unless of course it's a meme, in which case, figure out the context of that meme and then roll people through, like bring people to your level of understanding through that context. That's why memes are actually a really great tool for communicating science, because like we get like this visceral emotion when we see SpongeBob doing a SpongeBob thing, and if you can find a way to communicate that particular emotion, you can kind of teach a lot. Again, not accurately, but for getting people to like a biochemical level of understanding, we have to take them through a lot of layers of abstraction. And so you need every tool you can possibly have. Even if you think, you have a more, even if you're like communicating medicine, like that can also be very scary when you're communicating very complex things that can, be very existential to the person you're talking to. If you can kind of level them up through those layers of abstraction while repeating your main idea, you can also make that a little bit more comforting and easier to understand. The more somebody understands something, the less annoying or terrifying or scary it is. Understanding is a really great way to take away negative emotions and help people not be and not be distracting or not be jarring. The last thing you want to do is jar your viewer. I don't want to just say there's a 7,800 RPM motor inside every single one of your cells that's powering everything you do. That doesn't mean anything, but that's still my main idea. ATP synthase rotates roughly at 7,800 RPM, and there's about 78 quadrillion ATP synthase complexes in any given human body, give or take a few quadrillion. So that's how you can kind of bring that back, and that also make it very sticky. So that is a very, very basic level of how we do life sciences communication. You always want to find the context of your ideas. You want to level your audience up through that context, and then you want to really just blow people away with whatever main idea you have, and then you want to connect that to something larger, connect it to a bigger idea outside of what you're talking about. If you're talking about photosynthesis, you can talk about the entire universe. If you're talking about hemoglobin, you can talk about the incredible dance that is being alive. If you're talking about telomerase, you can discuss, it's kind of mortality is kind of scary, but it's also kind of beautiful in a way, because you see just how hard your body is trying to keep you alive at any given moment, so on and so forth. And now that I've done an hour of talking, I'm going to take a quick breath. Good. And at that point, take some questions. So I appreciate you guys hanging out with me. Again, when you're asking life sciences communication questions, the answer to every single question is it depends. So if you ask a question, I'm going to say it depends, and then try to drag you through some context real fast. And I see people in the comments who are also bagging me on not even needing to take a drink of water. I'm honestly surprised. I haven't done this in a minute. Instead of talking in lecture format, I have been talking in 15-minute increments on my YouTube channel. So this is honestly mind-blowing. And what does it depend on? Well, it depends on a lot of other factors. Like there's dependencies all the way down, frankly. So either way, y'all, it's looking like we're more vibing in the comment section. Thank God. And when it comes to how much of this was improvised versus how much was practiced, it was all practiced. It's just that everything I like having an improvisational feel to what I'm doing. I have a pretty big script right behind my Discord that I was reading through, but I also want to always give off that character of, I don't actually know what I'm talking about. So like I can encourage people to do their own exploring, because when you're talking about something deeply subjective, you should not be coming at it like, I'm not going to come at the anatomy of a good science video like somebody would be talking about, like the human heart. The human heart, there's objective muscle fibers, there's objective actin and myosin, there's objective ventricles and atria, and so on and so forth. When it comes to communication, there's too many variables. Like communication is a quantum system almost, because there's 6 billion, 7 billion, there's a lot of billions of people in this world with a lot of different perspectives. And so it is impossible to objectively discuss communication. So that's why I kind of like have this improvisational feel to it, because the main goal here is to speak to the difficulty of communication. Because if I come here and I'm like, I'm an authority, you're going to immediately want to like talk the way I do, and that's not going to be a good way to become a better communicator of your own ideas. And I understand, like I advertised that my TA was sitting here with me the whole time. My cat Alice, she has been, but she's decided to be very quiet. I'm sorry that we did not get a lot of cat action during this particular conversation. I'll make up for that, Catholic as I am, by posting pictures in the photos and videos section of BioCord throughout the rest of the week. Like I said, my brand's hella Catholic. And when it comes to the actual, so a lot of people are asking questions about the process for, what is it called? Clockwork itself. Like, how do you come up with these? Constant rewriting. I'm gonna level with y'all. I have been writing this hemoglobin script since 2007. What is it? 2020 now. I was sitting there in my introductory Bio 101 class at a liberal arts institution somewhere in Pennsylvania, and my professor was just like, "Yeah, hemoglobin, it changes its shape due to pH. I don't know. Let's move on." And I was like, "What? No." No way. And I spent, obviously, like, I wasn't working on it the whole time, but the whole deal with anything, when you're thinking about how to write something, you either live to write or write to live, and I'm very much in the latter category. I'm constantly finding a different way to communicate different ideas so that I can communicate them effectively. So when it comes to my process, it's constant revision because when you are writing a video, right, you write the script and it looks nice, and then you immediately change its context by speaking it out loud. And immediately you see a thousand different ways in which the writing does not work. And then you put in an animation, you realize you're over-explaining one bit or under-explaining another. And so when it comes to process, I write a script, I animate the script, I change the script, I then record the voice of the script, and then I put it all together, and then I cry. I cry so much, and then I pull an all-nighter, like 4 of them, and then I post the video, and then I fall asleep for 2 weeks. And then I come back to feedback. That's the clockwork process. Write, cry, write a lot, cry some more, and then rely entirely on BioChord. I'm not going to lie to you guys. This channel was launched 2 weeks after BioChord, completely coincidentally, and BioChord and Clockwork kind of came up together on the Reddit. Obviously, BioChord is much larger than Clockwork until you guys subscribe. Look, it's right there. Just do it. Hit that subscribe button, hit that bell, check out that Patreon. What was I saying? And so what it comes down to is, like, that's why having a community like BioChord is so valuable, because when you're communicating, just take your ideas and throw them into BioChord. There's 5,000, 4,000, there's a whole bunch of thousands of people in here just, like, ready to help you out. Look at all these beautiful people ready to help you out with anything you're working on, as long as it's in the right channel. Go to Lobby 2, Lobby 3, or Homework Help, and you have access to some of the most brilliant people on Earth when it comes to life sciences. I cannot tell you, like, if you are watching this on YouTube and not in the actual BioChord itself, drop everything you're doing, discord.gg/biology, get in here. The only way you really win in life sciences is if you have a community and if you have the ability to ask questions, because there's always another layer of abstraction. Eventually, you're going to get all the way to quantum physics and calculus if you really want to get down to the essentials of whatever you're trying to figure out in life sciences. And sometimes you have to just give up and understand, I'm not going to understand everything here, but I'm going to get as close as I possibly can. And the way you do that is by having an amazing community like the one that exists here in BioChord. Not to simp for BioChord too hard, but I gotta get in there real quick. I'm a YouTuber. Everything I do is ads. What do you want from me? All right. So when I think about my favorite episode of anything I've produced on so I'm gonna just go through questions. I have 3 minutes left. Let's see. So when you if I have to advertise my channel, if you want to know my favorite episode of a script I've produced, I would definitely just tell you to go to my channel, go to playlists, and just check out my photosynthesis playlist. I have one on photosystem 2, I have one on the plant Q cycle, and then on this Tuesday I will talk about how ATP synthase completes the light reactions of photosynthesis. This whole year I've basically made photosynthesis the movie, that's been extremely just like amazing and fun, and just like connecting this biochemical process to like the universe, and just how life is just this sort of like defiance against thermodynamics and all of that. Anyway, so that would be what I would say. Check out this photosynthesis playlist, and if you can appreciate the sort of breathless and overly enthusiastic style with which I do this sort of stuff, that's great. Let's go through. I'm gonna roll through more questions. Any tips for writing scripts? I have so many. The number one rule in life, the number one rule in any kind of writing, is to imagine your audience and try to find a way to use their time in a way they do not feel like it was wasted. Like, when I thought about making this presentation, I knew I had to be a little bit more entertaining than knowledgeable, because I didn't want to come off as a sort of like dry authority, since it's a very subjective material. So I'm hoping that I didn't waste anybody's time, and I'm hoping that I made this kind of fun as opposed to like cut and dry. And so if you appreciated the improvisational approach to this, I win. If you didn't, I lost, and I'll figure out how to find a better balance. Next question: is biology a real science? Biology is the only science. Everything else is made up. Moving on. BioCord is not paying for this promo, other than the fact that like I am partnered with BioCord. It's a loose. I mean, it's there's no we don't have it's not a legally binding document. ANS isn't going to come over here and be like, you said you do these things. But it's just that my channel would not exist without BioCord. Period. End of sentence. It wouldn't be any good. It would just be me flapping my gums about bad concepts. Everything good about my channel comes from people on BioCord telling me where to find free scientific papers, for instance, which I'm not going to tell you about here. But yeah, that's essentially it. Either way, we are at a hard out here. If you have any specific questions about writing, animating, anything, music production, audio, microphones, the whole works. I have been doing this for years. It's not just one YouTube channel. I have been animating and doing everything else for my entire professional career, and I've been teaching. My entire life has been revolving around teaching science and talking about science to folks who want to understand it. So I'm here on Discord. I'm Clockwork Show number something. Look for Clockwork Show. Hit me up here if you have any questions about how to communicate. It's always going to come down to very personal circumstances. Either way, I really appreciate everyone's time. Thank you so much. I hope you have a great rest of your conference, and see y'all later. Peace.
Thank you so much for that awesome talk. So this concludes the second day of the BioCord Winter Conference. If you have not already, please subscribe to Glorious Clockwork on YouTube. This talk was also recorded, so I will be uploading that to our YouTube channel too. Thank you a lot, and we will resume the BioCord Winter Conference next weekend.